will learn what are the sequencing data formats which are FASTA, FASTQ, and why are they in the format sheltered. And we'll learn how to navigate the NCBI SRA database, and we'll move ahead with the FASTQ retrieval from NCBI SRA database and preparing the retrieved data for downstream analysis. By the end of this session, you will be able to locate, retrieve, and interpret real bacterial whole genome sequencing datasets from NCBI SRA. So moving ahead to basic Linux basics for bioinformatics. So why learn Linux for bioinformatics is what we learn in this slide. Because most of the tools run over Linux, and Linux handles large datasets better than Windows maybe, and Linux provides the interface to actually maintain everything in a system, and there are many tools available for Linux as well. And for NGS sequencing, mainly we use SRA toolkit, FastQC for quality assessment, and NCBI BLAST, SAM tools, everything we have it in Linux. And what are the I want some answers from the audience. Have you used Linux or any command line tools before? Please type yes or no. No, yes. Yes, many have the answers as no. Okay. If you have not used Linux or any command line tools, just don't worry. We'll start from the basics of Linux. So for example, a Linux file system is basically, it has a root directory. It has a root directory where which is the topmost directory in Linux, and a home directory where you store your personal workspace, and under the root directory you create folders which are of use for you, and you have very specific folders for everything that you do, and these are the common directories and their purposes which are used in Linux. For example, root is where it is the topmost directory, and home, it contains personal files, and in the tools files we have all the scripts and tools installed in the tools folder, and TMP is a temporary folder. So when always when working with large sequence or any data that may be, when you are working with a large data, always keep your organization clean, so that when you revisit, you don't forget your folders and architecture of your folder, and just keep finding the file that you have kept somewhere. Yeah, always know your file where it is, and how where it is stored. So a simple idea is to store it in very specific directories. So some essential Linux commands include pwd, which shows the current or present working directory maybe, and ls, it lists the files in the folder, and cd is current directory, or you can use cd to move into the folder as well. mkdir is the short form for make directory, it creates a new folder, and head and for example, we have head sample FASTQ, which takes the first lines of sample FASTQ, and wget is the command to download the files from the internet. This is an example of a terminal. This is actually from my system. This is how our terminal looks. Why these commands matter are to navigate the folders and inspect the files, and easily download the sequencing data, and run the successful analysis. So where to practice Linux? If you are on Mac and Linux, there is already terminal available on Mac and Linux for Linux users. And so if you are on Windows, then then it may be a problem, not actually a problem, but you can download Ubuntu terminal on your Windows easily and practice Linux commands. So what is the operating systems are you using? Uh, from the audience, can you type in the chat what is the OS you are using? Windows, Mac, or Linux? Windows. Many are using Windows. Linux, Ubuntu. Someone is using Linux. Windows. So many are using Windows. Okay. For that we have I'll share it in the chat box, so you can actually download Ubuntu for Windows from the Microsoft Store. Sorry. Who all are using Windows, please have this link and download it and keep it ready for hands-on sessions. As many of you are using Windows, please download it and keep it ready. Next, we'll move ahead to what is NGS sequencing. In the Sanger sequencing, so for in order to first learn the NGS next generation sequencing, first we have to understand what is Sanger sequencing. In the Sanger sequencing, there is sequencing of DNA fragment, the DNA fragment at a time, and it is for small scale projects, like for example, if you have cultured a bacteria and you want to identify the bacteria, you go for 16S, and with it sequences only one bacteria, so it is enough. It is low, and it gives one read per, and it is high cost per base, but it is enough for if you want to sequence a single one. But when it comes to if you have isolated a microbiome, and when if you want to sequence using Sanger sequencing, it is very difficult, so we go for next generation sequencing, where the sequencer runs through the sample for multiple times, uh, and you obtain millions of reads per sample. And since the for a single sample you get many reads, and so the cost per base is very low here. And the difference between NGS and Sanger sequencing is, in the Sanger sequencing you get only one read per sample, and you get millions of reads per sample in NGS. And for Sanger sequencing, throughput is very low, and NGS it is high. Cost per base is high in Sanger sequencing, while in NGS it is very low. And whole genome sequencing is very difficult in Sanger sequencing. Sanger sequencing is applicable only in shorter sequencing, like for 16S RNA sequencing, and for whole genome sequencing we mainly go for NGS sequencing. So if a bacterial genome is 5 billion bases long, what kind of sequencing is needed to quickly sequence the entire genome? Audience, can you answer in the chat box? NGS, NGS. Yeah, many of you are right. Many of you are giving the right answer. So we'll move ahead since you understood the NGS sequencing. When it comes to whole genome sequencing, what is whole genome sequencing? We sequence the full genome. For example, in the 16S RNA sequencing, we only sequence the 16S rRNA. In the whole genome sequencing, we sequence all the chromosomes and all the DNA that is available in a particular bacteria or maybe a human genome. We go for bacterial cell isolation first, uh, and then DNA is isolated, and then we go for NGS, and genome reconstruction is followed. So application of whole genome sequencing includes pathogen identification, antimicrobial resistance, outbreak investigation. For example, we went for COVID-19, and comparative genomics maybe for gut microbiome and microbiome research as well. So if two bacterial isolates looks all identical in the laboratory, how can we determine whether they belong to the same outbreak? This is the question for our audiences again. Yes, whole genome sequencing. Yes, you are right. We go for uh, we go for whole genome sequencing. We'll have a look at overview of NGS workflow. First in whatever NGS we do, the first step is to collect the bacterial isolate or the biological sample, maybe a gut microbiome or maybe soil microbiome we can take, and we go, and then next step is the DNA extraction. A high quality genomic DNA is isolated, and then library preparation is followed after DNA extraction, and then it is fed into an NGS sequencing machine where millions of DNA fragments are obtained, and uh, millions of sequences are obtained in the FASTQ files. Then next it is QC, where quality is analyzed, and low quality reads are removed in the QC. And next is genome assembly and variant analysis. And next we go, it is biological interpretation. So again, question for audience: which step generates FASTQ files that we download from NCBI SRA? Yes, can anyone answer in the audience? NGS sequencing. Yes, NGS sequencing. Someone is answering QC. QC is after which we after we get the after we get the fast Q sequence, we go for QC. Not before NGS sequencing. After sequencing, we get raw data. Yes. You're right, Tamil. Yes, most of you are right with the answers. We'll go for FASTQ files. What is a FASTQ file? The FASTQ file is obtained once the sequencing is done. A FASTQ file basically contains sequence plus quality information. So, a sequence plus quality information. So, a FASTQ file consists of 4 lines. First line is the read identification. Second line is the DNA sequence, and the third line is the separator, always added by always represented by a plus sign, and line 4 is the quality scores. So one single sequence is 4 lines in FASTQ file, which is repeated million times if it is a NGS sequence. So we have answer in the slide. Which information is present in the FASTQ file? Answers from the audience, please. DNA sequence, yes. Sequence, right, but it contains 4 lines. Who is going to answer all the 4? DNA sequence and quality score, yes, right. You are very much right, and we'll move ahead to FASTQ quality scores. What is meant by FASTQ quality scores? For example, we have already learned FASTQ, an example of FASTQ sequence where first line consists of read identification identifier, next line is the DNA sequence, third line is the separator line, and fourth line is the quality scores. So the character I represents high quality base, and the other characters represents different maybe low quality basis. And what is pH red scores for pH red scores, which is represented by Q, is a way to express probability of an incorrect base call. For example, when we go for once we get the fast Q, we go for QC analysis, where we get the pH red score, and Q10 means 1 in 10, which means 0.1% probability that the database is wrong. For example, in the example I have mentioned here, for the first sequence, first base is A. There is 0.1% chance that A is wrong. That is meant by Q10. When it is Q20, it is 1 in 100 is 100. 1 in 100, there is 1 in 100 chance that the base is wrong. And Q30 and Q40 are usually considered as high quality data. Okay, when a sequencer reads the DNA, it can make mistakes. Everybody makes the mistake, and the quality scores help us determine whether the sequencing rates are reliable or not. For example, a sequence if a sequence read is taken, but there is very low quality scores, which may be due to preparation steps or library preparation step, or low quality DNA isolated before the sequencing. Maybe there are many reasons for sequencing to be of low quality. So we learn what is FASTQ and FASTA. In the previous training, we have learned what is FASTA, which consists of accession number and sequence identifier number and and sequence. It is only of 2 lines, whereas in FASTQ it is 4 lines. FASTQ will look at FASTQ versus FASTA. What are the key differences between FASTQ and FASTA? Both carry the sequence information, but only FASTQ carries quality scores, and FASTQ is raw read sequencing, and FASTA only contains genome assembly output. And in the FASTQ is the one we get in NCBI SRA, whereas in FASTA sequence we get from DNA sequencing portals such as from the NCBI itself, which is used in NCBI GenBank.